Now, let's get started with a review of the anatomy and physiology of the immune system. Unlike organs like the liver and heart, which are localized masses of tissue, the immune system is comprised of both tissues and cells throughout the body. The tissues are organized into primary lymphoid tissues, such as the bone marrow and thymus, that contain lymphoid progenitor cells and are involved in lymphoid cell production and development. There are also secondary lymphoid tissues, such as the spleen and lymph nodes, which play a role in lymphocyte homing, antigen presentation, and acquired immune response. The bone marrow contains pluripotent hematopoietic stem cells that are the source of most cells in the immune system. The thymus plays a later role in immune system development in that T cells produced in the bone marrow must travel to the thymus to complete their maturation. The thymus is an encapsulated lymphoid organ where T cell maturation and differentiation occur. Its cortex is rich with immature T cells, while the medulla contains sparse, mature T cells, epithelial reticular cells, and Hassel's corpuscles. A lymph node is an encapsulated, trabeculated lymphoid organ that has many afferent and one or more efferent branches. It functions as a filter, as a storage and activation center for B and T lymphoid cells, and as a center for antibody production. There are three main components of the lymph node. 1. The follicle, which is the site of B cell storage and proliferation. 2. Is medulla, which is composed of medullary cords and sinuses acting as the filter. And 3. Is the paracortex, which houses T cells. It is important to note that lymph drainage for specific anatomical structures flows to certain primary lymph node sites. For example, the arm and lateral breast drain to the axillary lymph nodes. The stomach drains to the celiac lymph nodes. Duodenum and jejunum drain to the superior mesenteric lymph nodes. The sigmoid colon drains to the colic and then to the inferior mesenteric lymph nodes. The lower rectum and anal canal above the pectinate line drain to the internal iliac lymph nodes. The anal canal below the pectinate line drains to the superficial inguinal lymph nodes. The scrotum also drains to the superficial inguinal lymph nodes, while the testes drain to the para-aortic lymph nodes. Anatomic structures of the spleen include sinusoids, which are composed of elongated channels with a fenestrated membrane and adjacent macrophages functioning as a filter. The periarterial lymphatic sheath, or PALS, in red pulp, which houses T cells, and follicles in white pulp, which house B cells. The immune system itself is divided into two types: innate and acquired. Innate immunity includes the physical and biochemical barriers both inside and outside the body, as well as immune cells that have specific responses. It has no memory and the innate immune response does not involve antibodies. In contrast, acquired immunity is the specific response to infectious agents that depends on the antigen-antibody response. The acquired immune system has memory and involves antibodies. Now let's take a closer look at these two branches of the immune system. Innate immunity is a nonspecific immune response that will indiscriminately attack invaders regardless of their nature. Innate immunity has no memory, which means that repeated exposures to the same pathogen will produce the same response as the first exposure to that pathogen. The innate immune system is the body's first line of defense. Most infectious agents enter the body through the respiratory system, gut, or genitourinary tract. When it comes to the innate immune system, the skin is an effective physical barrier against most toxicants. Other defenses include mucus secreted in nasal passageway that trap and sweep away particles and small organisms, cilia lining the trachea and main bronchi that also act to catch and sweep away infectious agents, and reflexes such as coughing, sneezing, and elevation of body temperature that serve to remove and destroy infectious agents. There are two primary cell types involved in the nonspecific innate immune response. The first is the natural killer or NK cell. NK cells recognize virally infected and malignant changes on the surface of cells, as well as antibody-coded target cells. When NK cells find such a target, they will bind to the cell and expel cytolytic granules that cause the target cell to die by apoptosis. The second type of cell is the professional phagocyte, which includes both polymorphonuclear cells, also called neutrophils, and macrophages. Phagocytic cells act to engulf debris, infectious particles, and bacteria, and destroy it to remove it from the body. Neutrophils are highly efficient phagocytes that can eliminate most microorganisms and induce an inflammatory response. However, if the neutrophils cannot contain the infection, then macrophages are recruited to the site of the infection to aid the process. In the innate immune system, the immune response is a broad-based defense mechanism that attacks pathogens in a sweeping, nonspecific attack. Innate immune system responses include complement. Complement is a set of plasma proteins that work together to destroy cell membranes, attract phagocytes, and stimulate activity of other immune system cells. Complement proteins are usually inactive. However, contact with microbes or with specific antibodies can trigger the proteolytic cascade that destroys the pathogen. Fever. One of the most common physiological responses to infection is fever, which is an elevation in body temperature. Although historically fever has been considered harmful, and long period of excessive fever can cause further damage, it is now recognized that fever plays an important role in helping the body defeat infections. While the mechanism underlying this benefit is not completely understood, some studies suggest that some immune functions are enhanced during fever. Fever is triggered by the release of pyrogens that increase body temperature, by inducing cyclooxygenase 2, or COX-2, an enzyme that produces prostaglandin PGE2, which triggers the fever response in the brain known as the preoptic area of the hypothalamus. Fever can be reduced by administering antipyretic drugs, such as aspirin, acetaminophen, or other nonsteroidal anti-inflammatory drugs, or NSAIDs. NSAIDs primarily act by inhibiting or downregulating the cyclooxygenase enzymes. And finally, inflammation. When tissues become injured, the typical response is inflammation, a series of responses that is initiated by mast cells embedded in connective tissue that release a number of pro-inflammatory cytokines and chemokines, including histamine and leukotrienes. These agents cause local blood vessels to dilate, which allows for an influx of immune cells to clear the area of pathogens and remove any cellular debris. The increased blood flow is usually accompanied by the typical signs of inflammation, which include pain, swelling, redness, and heat. In contrast to innate immunity, acquired immunity involves immune responses that are specific to the invading pathogen, and it does have a memory, such that repeated exposure to the same pathogen will produce an enhanced response to that same pathogen the next time it is encountered. The acquired immune response relies on the recognition of antigens by antibodies. An antigen is a substance that can elicit the production of a specific antibody and can be specifically bound by that antibody. Antibodies are proteins that are classified as immunoglobulins, are produced by B cells, and are defined functionally by the antigens to which they bind. There are 5 types of immunoglobulin that are structurally related: IgM, IgG, IgE, IgD, and IgA. The components of the acquired immune system can be divided into 2 categories: cellular and humoral. The cell-mediated component relies on direct contact between immune cells and the pathogen, while the humoral component is an antibody-mediated response that relies on antibodies to convey signals between cells of the immune system. Both B cells and T cells play important roles in acquired immunity. B cells and T cells differ in their functions. B cells make antibodies, while T cells provide delayed cell-mediated hypersensitivity. An important component of acquired immunity is the major histocompatibility complex, or MHC, that is encoded by human leukocyte antigen, or HLA, genes. MHC1 is encoded by HLA-A, B, and C. It is expressed on most nucleated cells. The antigen is loaded in the rough endoplasmic reticulum of largely intracellular peptides and mediates viral immunity. MHC2 is encoded by HLA-DR, DP, and DQ. It is expressed only on antigen-presenting cells. The antigen is loaded in an acidified endosome and aids in T cell communication with B cells and macrophages. You can remember this with the mnemonic that MHC1 is 1 HLA letter, A, B, or C, while MHC2 will have 2 HLA letters, DR, DP, or DQ. T cells play an important role in cell-mediated acquired immunity. T cell precursors from the bone marrow travel to the thymus, where they differentiate into CD8 plus cells and CD4 plus cells. These cells then travel to the lymph nodes, where they further differentiate. CD8 plus cells differentiate to cytotoxic T cells, while CD4 plus cells differentiate to helper T cells. Helper T cells differentiate further into Th1 and Th2 cells. T cell glycoproteins that are important components that facilitate binding to target cells include CD8 and CD4. CD8 is found on cytotoxic T cells and binds to MHC1 on virus-infected cells. CD4 is found on helper T cells and binds to MHC2 on antigen-presenting cells. T cells require two signals for their activation process, signal 1 and signal 2. For Th cell activation, a foreign body undergoes phagocytosis by an antigen-presenting cell. Then, signal 1 occurs when the foreign antigen, now present on MHC2 of the antigen-presenting cell, is identified by the TCR on the Th cell. And signal 2 is a co-stimulatory signal as B7 and CD28 interact. This activation allows the Th cell to now produce cytokines. For Tc, or cytotoxic T cell activation, signal 1 occurs when endogenously synthesized proteins are presented on MHC1 of the virus-infected cells and identified by TCR on the Tc cell. And signal 2 occurs when the Th cell interleukin 2 activates the Tc cell to destroy the virus-infected cell. Now let's take a look at the role of B cells in the immune response by taking a closer look at both the primary and secondary antibody responses. The body produces B cells that express a wide variety of surface receptors that recognize an array of antigens. Once a B cell encounters a specific antigen that is recognized as non-self, through the binding of the antigen to the appropriate antibody on the B cell plasma membrane surface, the B cell becomes sensitized. Further contact with T helper cells, which are cells that present antigen and secrete lymphokines, activates the B cell, causing it to proliferate and differentiate into plasma cells that secrete additional antibodies. The antibodies bind to any antigens with the same epitope as the antigen that elicited the original response, causing the antigen to become immobilized and marking it for destruction by phagocytes. After the invader has been neutralized and removed, antibody production ceases, and some plasma cells remain dormant in the body as memory B cells that remain dormant unless a later exposure to the same antigen occurs, thereby eliciting a secondary response. This entire process is known as the primary response. In the secondary response, the presence of antigen-specific memory B cells in the body allows the immune system to respond rapidly and at a greater magnitude during subsequent exposures to the same antigen. This is a direct result of the production of memory B cells, which are present in greater numbers than the originating B cells and can rapidly secrete antibodies when they come in contact with the appropriate antigen. Memory B cells can live in the body for decades, allowing the body to remember prior exposure to antigens for a long time. The secondary response is the basis for vaccination, whereby the body is exposed to an antigen that has been altered to be non-pathogenic or to a non-pathogenic antigen that is similar to the antigen of concern, but creates memory B cells that can mount a rapid response of greater magnitude than the primary response against the antigen. It's important to note that the primary response to an antigen is mediated by IgM and takes place 3 to 5 days after the initial exposure. When a second exposure to the same antigen occurs, B cells undergo isotype switching and produce IgG rather than IgM. IgG is of higher affinity to the antigen and is produced at higher concentrations compared to IgM. We've spent the last few minutes discussing antibodies. Let's take a closer look now at antibody structure. The Fab or FAB fragment of an antibody binds to antigens with the variable components of the L and H or light and heavy chains recognizing different antigens. The FC fragment of IgG and IgM fixes complement. There are three primary functions of antibodies: opsonization, neutralization, and complement activation. Opsonization is the process whereby the antibody promotes phagocytosis. In neutralization, the antibody inhibits bacterial adherence. And in complement activation, there is promotion of opsonization. Antibody diversity allows the body to respond to the presence of a variety of antigens. Antibody diversity is produced by random recombination of light or heavy chain genes, random combination of light with heavy chain genes, somatic hypermutation, and nucleotide addition to DNA during recombination by terminal deoxynucleotidal transferase. Now, as previously mentioned, there are 5 different isotypes of immunoglobulin. IgG is the most abundant antibody. It is a significant player in the secondary antibody response, and it acts to fix complement, opsonize bacteria, neutralize viruses and bacterial toxins, and IgG does cross the placenta. IgA is found in secretions. It prevents attachment of bacteria and virus to mucous membranes. IgM is generated in the primary response to an antigen, fixes complement, and does not cross the placenta. IgD is found in serum and on the B cell surface. However, its function is unknown. And IgE is the least abundant antibody. It is responsible for immediate hypersensitivity by causing release of mediators from mast cells and basophils after exposure to an allergen. No discussion of antibodies is complete without a discussion of epitopes. An epitope is the antigenic determinant or the specific macromolecule that is recognized by the immune system. Different types of epitopes include allotype, Ig epitope that varies among members of the same species; isotype, which is an Ig epitope common to a specific class of antibody, for example, IgG or IgA; and idiotype is an Ig epitope specific for a certain antigen. Now let's take a look at a few other components of the immune system, including cytokines, cell surface proteins, and complement. Cytokines refer to signaling molecules secreted by components of the immune system that facilitate cellular communication by carrying signals between cells and having an effect on them. There are a number of important cytokines secreted by different components of the immune system. For example, interleukin-1 is secreted by macrophages and causes acute inflammation. Interleukin-2 is secreted by Th cells and stimulates cytotoxic and helper T cell growth. Interleukin-4 is secreted by T helper 2 cells and encourages B cell growth, while interleukin-5, also secreted by Th2 cells, encourages B cell differentiation. Gamma interferon is secreted by Th1 cells and stimulates macrophages, and tumor necrosis factor, or TNF, is secreted by macrophages and mediates septic shock. Now let's take a look at cell surface proteins. Immunologic cells have certain cell surface proteins associated with them. Helper T cells have CD4, TCR, CD3, CD28, and CD40L. Cytotoxic T cells have CD8, TCR, and CD3. B cells have IgM, B7, CD19, CD20, CD21, CD40, and MHC2. Macrophages have MHC2, B7, CD40, and CD14. Finally, NK cells have receptors for MHC1, CD16, and CD56. The complement system consists of a number of small proteins in the blood that interact with the immune system to play a role in humoral immunity and inflammation. Complement is activated via two pathways, the classic and the alternative pathways. The classic pathway requires antibodies to be present for activation, typically IgG or IgM. The alternative pathway requires the presence of antigens for activation, such as an endotoxin. Interferons interfere with viruses by causing uninfected cells to have antiviral qualities. The types of interferon are alpha interferon, beta interferon, and gamma interferon. Antigen variation describes a process in which an infectious agent alters its surface proteins so as not to be detected by the host immune system. This may be accomplished by DNA rearrangement or RNA segment reassortment. Some classic examples of pathogens capable of antigen variation are Salmonella and Neisseria gonorrhea bacteria, influenza virus, and trypanosome parasites. We conclude this section with a discussion of anergy. Anergy refers to the lack of a host immune response when the host is presented with a known antigen. Anergy can occur at two levels. 1. T cells that are ordinarily self-reactive become non-reactive in the absence of co-stimulatory molecules. 2. B cells can also become non-reactive, but generally less so than T cells.